Hello. Thank you for viewing Equipping Payers to Outsmart Cancer with Game-Changing Healthcare AI Technology. Delivering on the promise of precision medicine, today. My name is Rob.
And I'm Gail, and we will be your presentation narrators.
This presentation satisfies the requirements for continuing education credit, as established by the International Society of Certified Employee Benefit Specialists. For 45 years, the highly regarded CEBS designation has delivered professional expertise, supported by research-based best practices. Established by the International Foundation of Employee Benefit Plans, and the Wharton School of the University of Pennsylvania, the CEBS program was designed based on the latest proven business ideas and tools, from world-acclaimed thought leaders. Today's presentation was prepared by Richard Nicholas, founder of the TPA Network Research Consortium, an emerging healthcare-focused nonprofit enterprise, created by industry veterans, to help payers evaluate new medical technologies and healthcare innovations, with comparative and translational research initiatives, conducted in real-world payer settings.
In this presentation, you will learn the cancer basics, in non-clinical terms, and the key oncology challenges that are facing employers and commercial payers. We will cover the important topic of comparative effectiveness research, and why reliance solely on the FDA and PBMs for guidance on medication selection is not advised. You will hear about new approaches to treating cancer, and how new technologies are being used to defeat it. And lastly, you will see how healthcare AI can be used in both clinical and payer settings, to improve outcomes, contain cost, manage utilization, and eliminate waste, by way of a deep dive real-world case study on CureMatch, a solution that harnesses genomics, artificial intelligence and a supercomputer, to power a comparative effectiveness approach to advance the fight against cancer. Here is a roadmap that provides greater detail about the topics that we will cover today.
Any discussion about how healthcare AI can be used to advance the treatment of cancer requires a basic understanding of the disease. As odd as it might sound, many people really don't know exactly what cancer is, or how it is caused or treated. So, if you find yourself in that category, you are hardly alone. Let's begin our discussion about cancer basics. Our body is made up of trillions of cells that form tissues and organs. Genes inside each cell instruct them as to when and how to grow, work, divide, and die. Normally, cells follow these instructions, but when this process fails, cancer develops. These errant cancer cells are called mutated cells, as they are no longer following the instructions given by the genes, and they continue to multiply and grow uncontrollably. Cancer is a general term to describe uncontrolled, excessive cell growth. When mutated cells divide and multiply, they can become a solid mass, called a tumor, or they can appear in the blood. When this occurs, the excess cells start to interfere with the body's normal functions. A cancerous tumor that continues to grow is called a malignant tumor. Cancer cells are very good at hiding from the immune system, and tricking it into helping them stay alive, grow or divide. As cancer cells don't stick together very well, they can spread to nearby tissues. When this occurs, the process is called metastasis. And lastly, with cancer, the organ that is initially impacted is often secondary in importance treatment-wise to the tumor's unique genomic makeup. This is because with cancer, no two are quite alike.
It has been said by CureMatch co-founder, and world-renowned cancer researcher and oncologist, Dr. Roselle Kurz-Rock, that cancer is like a snowflake, no two are quite alike. Following this analogy, do any of these cancers look alike? To reiterate, cancer is a general term for diseases that are similar but, like snowflakes, different. In the past, our approach toward treating cancer has been to prescribe the same medication to two patients, if they both have one similar mutation, even in the presence of different molecular profiles. Illustrated in the graphic at right, are the mutations for a patient with breast cancer, employing the old approach, in that two of the patients have the highlighted ERBB2 mutation. Both patients would be prescribed the same drug, despite the fact that the remainder of their mutations are all different. Understanding that all cancers are unique, the new approach toward treating cancer today is founded upon a consistent, science-based, molecular matching strategy, and having unfettered access to the full range of available cancer medications. Understanding that everyone's genomic makeup is different, and that the molecular composition of every cancer is unique, it only stands to reason that not everyone is going to react to a medication in a similar way. Simply put, medications do not have the same utility for everyone. The fact is, they are not intended to. As such, we should not expect them to act the same way for each of us. Yet, we do. At least as it relates to our traditional approach to the treatment of cancer. This begs the question, are we expecting too much from our drugs? When we think about medications, it is not difficult to misunderstand certain terms. By example, the term treatment effectiveness is used to describe a measure of the degree to which the treatment does what it is intended to do. Alternatively, treatment efficacy is the percent of the target patient population that derives a benefit from the treatment. A drug can be 98% successful at treating a disease, but for only 2% of its intended patients. This would evidence high effectiveness, but low efficacy. If a person were to know this information before taking a drug, he or she would have to feel pretty special, given the low odds of it working for them. Similarly, it is likely that people would act differently, if they really knew, and understood, the risk associated with taking certain medications. By example, the term, number needed to treat, refers to the number of people who need to be treated to help just one person benefit from the treatment. The term, number needed to harm, on the other hand, is the number of people who need to be treated for just one person to be harmed. How might you feel about a drug that requires 99 people to be treated before one person benefits, or a drug that needs only 20 people to take before one person is harmed? The truth is, the science is typically settled before a clinical trial actually begins, meaning that it is already known that the drug's mechanism of action actually works. As such, clinical trials essentially solve math problems, rather than clinical ones. They answer the question, what percent of the intended patient population actually benefited from the treatment? Again, the bottom line is that when it comes to treating cancer, like any illness or disease, the more medication choices that a doctor has, the greater the likelihood that the correct medication choice will be made.
We all know that in the general practice of medicine, physicians develop customized therapy combinations, or regimens, that are selected to simultaneously address each of a patient's specific medical conditions. By example, notice here that patients 1 and 2 both suffer from rheumatoid arthritis. Patient 1, however, also suffers from congestive heart failure and depression. Likewise, patient 2 also suffers from 2 other ailments, an infection, and hypertension. As you can see, while each has been prescribed the same medication to treat their rheumatoid arthritis, each patient was prescribed 2 other medications to treat their 2 other conditions. This is referred to as the patient-centric approach. Simply put, it is what we expect from our medical providers today. Interestingly enough, however, this is not the case as it relates to our approach to, and treatment of, cancer. Which begs the question, do we need a new approach to treating cancer? Let's take a look at where we are, and where we are going, as it relates to the evolving way in which we have been approaching the treatment of cancer. The graphic on the left illustrates our traditional treatment approach. The focus here is on finding a common feature among patients, and then placing all of them on the same drug. That common feature could be a type of cancer, a molecular aberration, a specific biomarker, etc. This is the drug-centric treatment approach. In this example, all 3 of the patients are prescribed the same drug as they all have marker A in common. The other markers are simply left unaddressed. The graphic on the right illustrates an evolved version of the drug-centric treatment approach, as practiced in the age of genomics. Because both of the patients have a common genomic mutation, marker A, they are both prescribed the same medication, despite the fact that the remainder of their molecular profile is entirely different. A detailed discussion of the topic of simultaneous versus sequential cancer treatment regimens is beyond the scope of this presentation. That being said, consider this. Your doctor informs you that your cancer was found to have 13 actionable mutations. She then asks, would you be okay with starting treatment to address 3 of them? How would you feel about that? Today, even among those who practice precision medicine, where very targeted drugs are used to address specific tumor mutations, some oncologists continue to follow a whack-a-mole approach, where a single drug is used to treat 1, 2, or maybe 3 mutations at once. Unfortunately, this approach leaves many potentially dangerous yet actionable cancerous cells unaddressed, and even enables them to develop resistance to certain drugs over time, thereby making them more difficult to defeat later. Here again, the bottom line is, if at all able, try to embark upon a cancer treatment plan that addresses as many tumor mutations as possible simultaneously. Today, we know to approach cancer with precision by using customized, patient-specific, combination drug treatments that are developed based on matching a patient's unique molecular and immune markers to all available on- and off-label drugs. This is called the patient-centric treatment approach, and as is easily seen, it greatly resembles the patient-centric approach that was detailed in our previous discussion about prescribing in the general practice of medicine.
Let's shift gears for a bit and step away from the clinical aspects of cancer to discuss the direct financial impact of the disease on payers. The facts are actually pretty grim. A person's chance of developing cancer is 1 in 200 each year, or more than 40% over a lifetime. In the U.S., cancer remains responsible for 1 in 6 deaths. Sadly, it is widely believed that many cancer patients get suboptimal cancer treatment, as most cancer drugs are selected on a hit-or-miss basis, rather than as a result of a scientific, or molecular, analysis. Indeed, fewer than 2% of cancer treatments are optimized via personalization, based on genomic information. As a result, as much as $150 billion annually is spent on high-cost, low clinical-value cancer drugs, that could be avoided with evidence-based controls. To complicate matters, medical knowledge is doubling every 73 days. Understanding this, how can oncologists keep up with all of the advances that are occurring? Cancer remains the most elusive condition facing payers, representing a full 30% of one employer stop-loss carrier's reimbursements for claims for its top 10 medical conditions. Remarkably, some 60% of plan sponsors will have at least one cancer shock loss claim each year, and, despite a lull in utilization due to the COVID-19 pandemic, cancer claims have been increasing in frequency, severity, and cost. As illustrated by the exhibit on the left, in 2020, 9 of the top 10 high-cost injectable drugs for this employer stop-loss carrier were for cancer. In fact, for this carrier, the average drug component of their cancer treatment claims was approximately $90,000. As illustrated by the exhibit at right, some 27% of the cost of cancer care was directly for drugs. It is interesting to note that some 80% of adverse drug events are due to a drug's interaction with a patient's genomic makeup. All of this suggests that cancer will continue to remain a major cost for payers, in large part, as a result of the ongoing development and release of new high-cost medications to treat it. As a result of our recent experience with COVID-19, all of us have become more familiar with the 3-letter government agencies that respond to events of this type, and, as is sometimes the case, they did not always live up to our expectations. For those in the healthcare sector, however, whose business it is to rely upon, and take guidance from, such agencies, it might be surprising to learn that their expectations of, and reliance upon, certain of these agencies, may not have been well placed. With this backdrop, let's examine what it is that the FDA does, and does not do. A survey of physicians recently revealed substantial deficits in knowledge, as it relates to the meaning of FDA approval. It indicated that some 70% of physicians believe that the FDA only approves new drugs if they are at least as good as existing ones, and that the FDA approves drugs based on clinically meaningful benefits. Unfortunately, neither is correct. To be approved by the FDA, a new drug must only show statistical significance on a primary outcome compared to a placebo in a small number of studies. Let's go over that again. To be approved by the FDA, a new drug must only show statistical significance on only one primary outcome compared to a placebo in only a small number of studies. Let's unpack this. Statistical significance means that there is less than a 5% probability that the results of a clinical trial happened by random chance. Clinical significance would be far more relevant. Only a single primary outcome is required, rather than the most important outcome. Comparing something to a placebo is proving that it is better than nothing. comparing the new drug to an active control, the standard of care, or even an existing drug, would be much more useful. And lastly, the FDA only requires a small number of studies to be provided as evidence. Interestingly, more than half of all breakthrough drugs are approved based on only one trial. The reality is that the FDA's low standard for obtaining approval does not include assessing how well a drug works. As a result of these misconceptions, doctors often overestimate the nominal evidence of efficacy required of new drugs, and then misprescribe, believing that the new drug is more effective than those presently available, when that may not be the case, thereby increasing payer costs.
Let's talk a little about the impact of breakthrough drugs. Several pathways exist to expedite the FDA approval of new drugs used to treat serious and life-threatening conditions. One way is to obtain breakthrough drug status, which can be granted if preliminary evidence indicates that the drug may demonstrate substantial improvement on a clinically significant endpoint over available therapies. The topic of breakthrough drugs is important, as nearly 60% of them, more than 125 by number, have been for the treatment of cancer. Despite the suggestion that these drugs need to provide more convincing evidence than ordinary drugs, a critical Yale study shows that breakthrough drug approval is based on shorter, smaller and fewer clinical trials, many of which lack randomization or double blinding. Some have even used surrogate lab tests as a substitute for real clinical endpoints. As a result, many physicians wrongly believe that breakthrough drugs are supported by stronger evidence than the FDA requires. This often leads to their being overprescribed. In fact, these misconceptions about the term breakthrough often leads patients to be overly optimistic about a drug's true efficacy and, unfortunately, lead doctors to select them ahead of others. Evergreening is the process whereby drug manufacturers tweak an existing drug with a new release form, different dosages, combinations or variations. This is largely done for the purpose of extending a drug's patent, and artificially high price, as typically the tweak creates no therapeutic advantage, or increase in efficacy. The facts are that some 75% of new drug patents are not for new drugs, but for existing ones. And 60% of the best-selling drugs over the past decade had their patents extended at least once. Evergreening may also endanger patients who participate in what are unnecessary clinical trials. Essentially, evergreening supports the development of high-cost, low clinical value drugs that represent great waste for payers and financial toxicity for patients. That the practice of evergreening continues to occur is clear evidence that we must revisit how it is that we assess a drug's value.
As it relates to sources of dependable medical treatment guidance, it is necessary to discuss clinical practice guidelines. Many doctors rely on clinical practice guidelines from professional organizations to help them with their complex treatment decision-making. By example, these guidelines often provide doctors with advice on which medications they should consider or use. It is noteworthy to mention that most clinical practice guidelines are created by consensus, that is, expert elicitation, collective opinion, or educated guess. The fact is that the evidence upon which most clinical practice guidelines are based is often not subject to an exhaustive systematic analysis. In fact, often, conflicts of interest among clinical practice guideline contributors can consciously or unconsciously influence their decision-making, thereby complicating matters further. Unfortunately, in practice, the clinical practice guidelines development process does not always support true, unbiased, and objective, evidence-based advice.
Because the cost of medication is such a large portion of the total cost of treating cancer, it is helpful to have a basic understanding of how the pharmaceutical industry operates, and the role of the pharmacy benefit manager. We have some credibility in this regard, as about 20 years ago, we authored some of the very first white papers in health economics studies that detailed the then largely unknown inner workings of the drug industry, with the assistance of one of the federal government's leading forensic PBM auditors. Since then, the pharmaceutical industry has evolved and said to have adopted a more transparent, efficient, and cost-effective business model. Today, the industry is committed to achieving the best clinical outcomes at a fair cost, by supporting patients, pharmacists, doctors, and payers alike. Pharmacy benefit managers, also known as PBMs, emerged in earnest in the 1980s when they enabled health insurance companies to extend medical coverage to include drugs, by automating pharmacy claim processing, thereby making it cost-effective. Over time, PBMs morphed to represent the interests of payers and plan sponsors in negotiations with Big Pharma. They then began to manage the drug formulary and plan design, ostensibly to improve patient quality and reduce payer costs. Today, pharmacy benefit managers are an integral part of our healthcare system. Indeed, the top 3 PBMs control some 80% of the drug market. Healthcare observers and critics agree, the U.S. healthcare system is broken, or at least not operating as efficiently as it should. In large part, critics point to the fact that in many cases, payer, patient and pharmaceutical industry alignment is not in place. They argue that our pharmaceutical system will only work when everyone's incentives are realigned. Understanding this, it is important to underscore the role of PBMs, as they heavily influence drug access, prescribing and dispensing, with their e-prescribing, network, formulary, and prior authorization processes. Collectively, the 3 largest PBMs own mail, retail and specialty pharmacies, that dispense more pharmaceuticals than any other entity. Given that some 80% of PBM revenue comes from drug sales, they benefit directly from the volume of drugs sold. Therein lies the essence of the misaligned incentives criticism. As a result, as you will see later on, these misaligned incentives can cause payers, often unknowingly, to design their drug plans and formularies in ways that disadvantage them and their plan participants. One way by which plan sponsors and payers unwittingly disadvantage themselves, is by chasing what is called the lowest net drug cost, at the expense of promoting the most appropriate and effective drug. It is well known that nothing about the way in which prices are established, or suppliers reimbursed, makes much sense in the pharmaceutical industry. One such example involves the recently publicized industry practice of rebating. Manufacturers pay PBMs large rebates on brand drugs, in order to influence the placement of a particular drug on the PBM's formulary, in a favorable tier position, with a lower copay, or other advantage. This is called formulary placement. Rebates, which essentially resemble kickbacks, can be substantial, as much as 40% of the manufacturer's list price. As such, this model is meant to favor high-cost versus low-cost drugs, where the rebates are likely to be more substantial. Unfortunately, these drugs are not necessarily the ones that are the most effective, or the best value. Astute payers negotiate to have all rebates passed on to them. Though it is estimated that few payers get more than 40% of the amount paid to the PBM. Believing that they are striking the best deal, payers then often go on to design their drug plans, or enter into PBM contracts, that work to their disadvantage. The bottom line is simply this: creating a formulary to optimize net drug cost, by limiting placement to only those drugs that pay comparatively higher rebates, is both clinically and financially unsound policy.
As if coping with potentially biased information and guidance from presumably reliable resources is not enough, payers and plan sponsors also tend to rely upon approaches, methods and tools to assess value that are unreliable or misleading. As such, distinguishing between waste and value has become more difficult than it sounds, especially as it relates to assessing the value of high-cost specialty medications, in a drug industry that doesn't operate like other free markets. Is comparative effectiveness research and analysis the solution? Let's review a case study to illustrate how it may be the answer to this problem. This case study involves anti-clotting drug thinners. Here, drugs are measured and compared as to their effectiveness, on the horizontal axis, and the incidence of associated side effects, on the vertical axis, as this is a critical factor with regard to these medications. Drugs that appear in the red zone are less desirable as they have more side effects and are less effective, whereas those in the blue zone are more desirable, as they are both more effective, and have fewer side effects associated with them. The standard of care is found at the center of the exhibit. Trade-offs must be made when considering drugs that fall into the other two gray zones. Essentially, this is a story that illustrates how it is that both sophisticated and unwitting buyers can fall victim to the practice of formulary manipulation, and the rebate-related net brand pricing fallacy. Each of the 7 drugs shown in the illustration treat blood clots in legs. Only the 3 highlighted drugs, however, are on the PBM-designed formulary, and thereby covered by the health plan. 4 drugs were purposefully left off the formulary. Note that the drugs in blue are brand drugs, and that those in red are generics. Drug G is the standard of care. This illustration demonstrates how adding drug cost data exposes high-cost, low clinical value drugs. Drug A brand drug, is about the same cost as the standard of care, drug G, yet it is far more effective, and causes far fewer side effects. Drug B, a generic, is less effective, causes more side effects, and costs an astounding 8 times more than drug A. This supports the notion that a drug's cost tells you nothing about its clinical value. It also raises several questions. Why is expensive drug B on the formulary, with a low generic drug copay, and low-cost high clinical value drug A on the formulary with a high brand copay? Worse yet, among all these medications, high-cost, less effective drug B was the last to be FDA approved. Given the other more effective, less costly drugs on the market, does this make sense that this high-cost, low clinical value drug be added to the formulary? Only though comparative effectiveness analysis is this clear, despite it being counterintuitive.
Let's pause for a moment and assess where we are. Cancer claims are increasing in frequency, severity and cost. New cancer drugs are expensive and not always better. Medical knowledge is increasing faster than humans can uptake and process it, at least without machine assistance. Confusion about FDA approval and breakthrough drugs often results in faulty prescribing assumptions. Industry consolidation and unaligned payment incentives confound the free market operation of the drug industry. And many of the traditional methods to assess the cost-effectiveness, return on investment, and value of drug treatments are unreliable, misleading, and obsolete. All in all, the deck appears to be stacked against payers.
Actually, it is not as bleak as it appears. There are many new medical technologies, healthcare innovations, and approaches that are available to help payers gain control over their cancer risk. Today's presentation will cover each of these topics: next-generation DNA sequencing, precision or personalized medicine, genomic-based targeted treatments, novel multi-drug combination cancer therapies, off-label drug use or repositioning, advanced clinical decision support systems, and healthcare AI. We will tie all of this together in our deep dive case study on CureMatch.
Next-generation sequencing, also known as NGS, is a revolutionary new form of DNA analysis that has changed cancer testing and treatment. Commercially available for clinical use only since 2005, NGS enables the reading of the full human genome to fully determine its unique molecular code, identify hundreds of biomarkers and mutation drivers, and permit the interrogation of normal and abnormal genes. NGS provides the basis upon which a genomic profile of a person, or tumor, can be created. As such, NGS supports the practice of precision medicine, and enables the design of tumor-specific cancer therapies.
Precision medicine, also known as personalized medicine, refers to any set of strategies that use a patient's unique genomic information to guide clinical actions that are tailored specifically to them. Precision medicine is based on the premise that the right treatment, provided at the initial discovery of a disease, will lead to quicker, better, and more definitive outcomes. Proponents believe that precision medicine will result in lower total long-term healthcare costs. In large part, this is owing to fewer and less resource-intensive diagnostic episodes and therapeutic regimens. Off-label drug use, also referred to as drug repositioning, is a therapeutic approach whereby current drugs, that are primarily used to treat one indication, are used to treat another. When treating cancer, drug repositioning refers to the use of FDA-approved drugs in combinations that are intended to address gene mutations in novel ways, as there is a greater likelihood that a novel multi-drug treatment will yield a better outcome the first time, versus repeated attempts using ordinary drugs. It is believed that between 50% and 75% of all chemotherapy agents are prescribed off-label. Typically, but not always, combination drug therapies involve the use of at least one drug as intended. The repositioning of cancer drugs expands the universe of available treatments enormously. Novel multi-drug or combination cancer therapy is a term that represents a treatment modality that combines two or more therapeutic agents to enhance efficacy, versus the single drug approach. Drug combinations are developed based upon the specific group of genomic markers that are being targeted. The results are often synergistic and/or additive. Combination therapies have been used to treat many diseases since 1965. One of the most notable of these were the multi-drug HIV cocktails that emerged in the early 1990s. FDA-approved combination drug treatments to treat cancer also date back to the 1990s. The use of the term novel relates to the unique combination of drugs that comprise the treatment regimen. Once again, it is the combination of the drugs that is novel. All of the drugs used are always FDA-approved medications. Combination therapies have been so successful that manufacturers are now researching and marketing pre-packaged combinations. One of the most highly promoted of these is Opdivo plus Yervoy. Both drugs are immunotherapies, used to strengthen the body's immune response to cancer, but in different ways. When they were found to work better when used together, the combination became a new product. It is important to note that cancer patients have a limit to the amount of toxicity that they can handle, and most of these drugs are toxic, to some degree. Also, many cancer medications have been found to work equally well, at a reduced dosage. As such, when drugs are used in combination, the dosages are typically cut by 1/2 to 2/3 for 2 and 3 drug combinations, respectively. Genomic-based targeted cancer treatments are exactly what they say they are. Treatments developed to target specific cancer cell mutations. The premise behind genomic-based targeted therapies is based on the recognition that each patient's tumor is unique, and that traditional, one-size-fits-all treatment is suboptimal in 2022. Because we know today that the molecular profile of a tumor mutation is as important as the organ of origin, we can now develop personalized drug treatments. These custom-designed, tumor-specific treatments leverage a tumor's unique characteristics to treat it. They do this by destroying targeted cells, changing proteins, and otherwise thwarting the uncontrolled growth of cancerous cells. Clinical decision support systems are computer-based health information technologies that are intended to improve healthcare delivery by augmenting clinicians in their complex decision-making. The term advanced is meant to distinguish those systems that utilize artificial intelligence. These platforms integrate and process patient-specific diagnostic and treatment data, targeted curated clinical knowledge, and intelligently filtered health information, to leverage knowledge, observations, analysis, and data that is not otherwise obtainable, or interpretable, by humans. Without such technology, the best of these systems demonstrate effectiveness and create guidance, advice, recommendations, and policy for users. Artificial intelligence is a term used to describe a variety of techniques that are intended to enable computers to mimic, or simulate, the problem-solving and decision-making capabilities of humans. When AI is employed in the healthcare industry, it is commonly referred to as healthcare AI. Investment in healthcare AI has exploded, with tens of billions of dollars backing companies that are expanding and driving a paradigm shift in how we both approach and treat diseases like cancer. Healthcare AI now enables us to translate complex data into comprehensible, actionable insights that help clinicians more accurately, quickly and effectively diagnose and treat cancer. Not surprisingly, AI is rapidly becoming the basis of advanced computer decision support systems. Numerous examples exist that demonstrate the promise of healthcare AI. Here are a few innovations that have already made it into practice. CureMatch is using healthcare AI to identify abnormalities and microscopic imaging changes, better and faster than humans. Viz AI helps to detect and identify misdiagnosed illnesses and medical errors. Olive's AI platform automates everything from insurer eligibility checks to unpaid claims for medical offices. Vicarious Surgical combines AI and virtual reality to enhance robotic surgery. The QVentus AI-based platform prioritizes hospital activity to benefit all patients. Tempus uses AI to distill the world's largest collection of clinical and molecular data to give doctors insight into personalized therapies. As these examples demonstrate, artificial intelligence is being used in a wide variety of medical delivery and healthcare administration settings, to drive knowledge, efficiency, and value. With a better understanding of what healthcare AI can do in practice, you can now see that it is fair to ask, is it possible that AI-supported comparative effectiveness research and analysis is a solution that payers can use to reduce their cancer risk and spend? As we have seen, the standard for a new drug to get FDA approval is fairly low, and not all approved drugs justify the high price set by their maker. This is especially so if the new drug is no better than existing drugs. This, coupled with the misplaced incentives and conflicts of interest that characterize the pharmaceutical industry, provide ample justification for payers to abandon the obsolete drug assessment methods that they have been using. As we have learned, comparative effectiveness analysis is undertaken, recognizing that we live in the real world, with existing drugs. As such, it looks at evidence as to how well a drug works versus other medications on the market. It considers active controlled trials, or employs a rigorous, scientific, meta-analysis, to get a full, realistic view of a drug's true effectiveness. All clinical trials and relevant drugs are considered, evaluated, ranked, objectively, without bias. Comparative effectiveness analysis answers the key question: does the data show a clinically significant difference, on a clinically meaningful outcome? To help answer this question, let's do a deep dive on CureMatch, a new healthcare innovation that actually leverages comparative effectiveness research and analysis, with the help of artificial intelligence.
Understanding that cancer is complex, and that every cancer is different, CureMatch leverages genomics, a curated knowledge base, artificial intelligence and a supercomputer, to unravel this complexity, to find and match the right drugs, to the unique molecular makeup of a patient's tumor. We will begin our discussion with an explanation of the clinical problem that CureMatch solves. To begin, there are more than 300 cancer drugs that have been approved by the FDA. These drugs generate more than 4.5 million 1, 2, and 3 drug combinations. CureMatch's curated knowledge base and AI-powered algorithm use a tumor's DNA profile to identify every actionable mutation, and the drugs that are likely to be best able to address them. An actionable mutation is one for which there is an anti-cancer drug that is available and known to target it. CureMatch analyzes all possible drug combinations and makes treatment recommendations that are based on a tumor's unique genomic profile, and those drugs that are most likely to produce higher progression-free, and overall, cancer survival rates. The analysis generates a clear, concise report that scores and ranks 1, 2, and 3 drug therapies that best target the cancer at a genomic level.
Here is how CureMatch works in practice. Once diagnosed, a tumor sample is collected, in either or both solid or liquid biopsy form. The sample is then sent to an NGS lab to sequence, and a tumor profile report is created. This report, in PDF format, is electronically sent to CureMatch, where the data and information are input. After a CureMatch analysis is performed, a process that typically about 48 hours, a full report is electronically sent to the treating physician for consideration.
Here is how CureMatch leverages artificial intelligence. The key input variables are patient diagnosis and age, the results of the NGS report and any other tumor pathology reports, any tumor immune profile, as well as other similar data and information, including pharmacogenomic test results that identify drug metabolism and drug interaction issues. This information is then considered in the context of the CureMatch proprietary knowledge base, which consists of manually curated evidence that includes clinical genomics, drug mechanisms of action, drug labels, clinical trial results and findings, etc. The CureMatch system identifies all actionable mutations, and all possible 1, 2, and 3 drug combinations, and then evaluates, scores, and ranks them, based on their ability to address as many of the identified actionable tumor mutations as possible. Clearly, given the sheer numbers involved, none of this could ever be possible without the assistance of artificial intelligence.
Let's dive a little deeper to learn why AI is able to do what humans are simply unable to, by looking closer at CureMatch's algorithm. Using the genomic input data, all actionable mutations are identified. In this case, there are 5 actionable mutations. Based on CureMatch's expertly curated knowledge-based content, it is determined that there are a total of 39 drugs that are able to target the actionable mutations. Note that of these, only 3 are on compendia, meaning, in practical terms, that ample evidence exists to convince expert reviewers to conclude that the off-label use of the medication should be approved for insurance coverage, and be eligible for reimbursement. This is extremely important, and very limiting, in that an estimated 50% to 75% of cancer medications are prescribed off-label. If CureMatch were only to consider combinations of on-compendia drugs, there would be only 7 options, 3 one-drug options, 3 two-drug combinations, and 1 three-drug combination. Here, you can see that by considering all of the 39 drugs known to address this tumor's 5 actionable mutations, the number of combinations increases from 7 to 9,919. CureMatch filters, scores, and ranks all of these combinations. However, only the top 3 1, 2 and 3 drug combinations are reported. As we have seen, by including FDA-approved drugs, on an off-label basis, to create its recommended novel combinations, CureMatch distinguishes itself from other precision oncology solutions that are severely limited by only considering on compendia drugs. In the example above, this would have restricted the universe of considered medications from 39 to only 3, and the number of considered drug combinations from 9,919 to only 7. Only CureMatch's patent-pending, proprietary technology can match many-to-many mutation combinations with novel multi-drug combinations to fully address each patient's unique molecular tumor profile. In this regard, CureMatch uniquely fills a very important, unmet market need. Here is the first page of a typical CureMatch report. It provides a snapshot of the recommended treatment options, in summary form. To arrive at this point, CureMatch identified, scored, and ranked 9,919 possible drug combinations. The Precis Match Therapy Matching Score represents the degree to which a given therapy option addresses a patient-specific molecular cancer profile. Physicians can input any drug, or drug combination, to be scored if they wish. Treatments that may be scored can include combinations of the standard of care, immunotherapies, chemotherapies, hormone therapies, etc. Likewise, treatments may also be excluded from scoring, including those that have failed in the past by example, and, of course, the CureMatch analysis accounts for any drug resistance, pharmacogenetics, and toxicity.
Focusing only on clinical results, let's look more carefully at this example. Note the scores generated by the 3 single drug recommendations. Trametinib, a targeted therapy known to be effective against mutations associated with skin cancer, scored only 17%. Meaning that it is likely to target only 17% of the identified actionable mutations. Cisplatin, a form of chemotherapy, scored only 22%. Pembrolizumab, known more popularly by its brand name, Keytruda, is an immunotherapy intended to support the body's own immune response. It scored 50% as a monotherapy. Notice that in combination, however, these drugs scored an impressive 70%. A detailed explanation as to why each of the 9 featured recommendations scored as they did, is provided later in the report. As shown, the genomic rationale for each recommendation is presented, along with references to supporting studies, reports, etc. For illustrative purposes, highlighted at right, are the sections of the report that provide details about 2 recommendation options, a 2-drug combination, and the top-scoring 3-drug combination. The targeting description shows exactly how each drug, in a mono or combination therapy option, addresses the cancer's known actionable mutations. To the right, it is noted whether the drug used is FDA approved for this indication, or, if it is being used, off-label. Note that the use of trametinib, in this example, is off-label. Drug contradictions, drug-drug interactions, and FDA black box warnings for each drug are provided, as well as a list of existing clinical trials where any of the combination's therapies is being examined. One or more of the prescribing physician's single or multi-drug treatment choices may also be scored. Many oncologists are cautious by nature, others are simply hesitant to develop novel combination treatments on their own. It is not surprising then, that the scores for the treatments that CureMatch scores for them are typically very low, often in the teens, sometimes in the single digits. Note, at right, that when available, clinically alternative drugs are identified. The substitution of these drugs does not adversely impact the combination drug score. This ability to consider alternative drugs can give a physician the flexibility needed to work around formulary restrictions, limitations and incentives.
To illustrate what all this means, in a real-world situation, let's consider 2 case studies, involving patients having different forms of metastatic liver cancer. Patient 1 is a 62-year-old man, who, after being diagnosed with liver cancer, was initially treated with chemoembolization that proved to be ineffective, as the cancer metastasized and spread. Despite subsequent therapy with sorafenib, disease progression continued, until a personalized medicine approach was adopted. A liquid biopsy was taken, an NGS test performed, and several actionable mutations were revealed. The patient was then cure-matched. The highest-ranked option was a 2-drug personalized combination therapy, using palbociclib and celecoxib, with each targeting specific mutations. The oncologist selected this option, and the patient's condition stabilized, as a result of the personalized approach. Patient 2, a 64-year-old woman diagnosed with liver cancer, was treated with chemoembolization. While awaiting a transplant, a year after the surgery, she received a gastrointestinal cancer diagnosis. She and her doctor then adopted a precision medicine approach. Both solid and liquid biopsies were taken, NGS performed, and several actionable mutations revealed. This data was integrated and put into the CureMatch platform. The highest-ranked option was a personalized, two-drug combination, using cabozantinib B and sirolimus, each intended to target specific mutations. Within a month, a lack of tumor growth appeared. As a result of this personalized combination therapy, the patient's condition stabilized.
One day, we are hopeful that everyone who receives a cancer diagnosis will be cure-matched, regardless of when on the cancer care continuum the diagnosis is made. Until that time, however, therapy matching is being restricted to use in advanced, largely third and fourth stage, cancer cases.
The scientific evidence to support the clinical utility of CureMatch is abundant and incontrovertible. Numerous peer-reviewed studies, published in leading scientific journals, show that with CureMatch a clinician is 350% more likely to select a more effective therapy option the first time. Indeed, it is noteworthy to mention that the creator of the scientific underpinnings of CureMatch, Dr. Roselle Kurz-Rock, has been involved with 850 peer-reviewed scientific papers. In fact, her work has been featured in the blog of the former director of the National Institutes of Health. And she was just with the president in the Oval Office, at the announcement of the new goals for the Cancer Moonshot. Dr. Kurz-Rock is also among the most cited scientific researchers in the world.
We have seen from the previous case example, how CureMatch can guide a physician away from treatments that are known to be ineffective, towards ones that have a higher likelihood of defeating a tumor's actionable mutations. Now, similar to the comparative effectiveness case study analysis that involved anti-blood clot medications, let's see how adding drug cost data greatly enhances payer utility, by enabling CureMatch to be used to reduce wasteful spending, in favor of treatments that represent a genuine financial value. The results of the case study we reviewed earlier appear on the left of the report. Added to the clinical results, at right, are the approximate costs of the various treatment options. In the first column, appears the full cost of each component drug, for 1 month. As drug dosages are reduced considerably when used in combination, the second column shows the 3-month cost of the treatment, after the dose adjustment. The third column is a comparative value index. It is calculated by dividing the 3-month adjusted dose cost by the number of CureMatch points scored. In this example, the full cost of 1 month of cisplatin, plus palbociclib, plus bevacizumab, is $27,689. After adjusting the dose, and the 3-month interval, the cost is $33,227 for 3 months of therapy. The matching score is 83% for this option. Therefore, the cost per CureMatch score percentage point is $400, which suggests a better value than the next best scoring option at $404. Note that the 2 FDA-approved, on-label single drug options for this indication, olaparib and carboplatin, each scored only 33%. Although these medications are popular, they represent a comparatively low clinical value option, when compared to other 2 and 3 drug alternatives, that are far better values. Accordingly, selecting either of these 2 drugs would be wasteful, given the other available options. As you can see, this index provides a good measure of the relative value of each option, at the point of care, a utility that could be of great value to a payer.
Here's another case study, illustrated in a new, slightly different, 2-column format. Note that the 6 top-ranked therapies scored 82% to 84%. The best value being the least costly of these. Also note the enormous difference in the cost between the 2 low clinical value options that scored only 17%, both of which are FDA approved for on-label dispensing, for this specific indication. In this case study, a 2-drug combination is the best value, with a comparatively high score of 74%, creating a significant financial savings opportunity, versus the top-scoring 3-drug options. Note that this 2-drug combination is also the second least expensive option. In this case study, the best scoring option, a 3-drug off-label combination, represents the best value, with a score of 62%. This option, at less than $18,000, creates a significant financial savings opportunity versus all of the scored alternatives. Note that each of the two low clinical value, single drug combinations, are far more costly. In March of 2022, the Research Consortium released a new study that challenges common misconceptions about combination cancer therapies, their cost and value, entitled, Gaining Control of Combination Cancer Treatment Risk, by Incorporating Cost and Value Data into the Drug Selection Process, at the Point of Care. The study was conducted using clinical case study data provided by the world-renowned cancer doctors and research scientists at CureMatch. This data was processed through the CureMatch system, and some 90 customized combination therapies were evaluated. Further, the cost-value index, described above, was applied. The study's findings are of keen interest to cancer patients, clinicians, payers, and manufacturers alike, as they provide insight as to how they can avoid wasteful spending on high-cost, low clinical value cancer drugs. The study noted that while it might be intuitive to believe that the cost of a treatment which involves the use of multiple high-cost drugs would be far more than the cost of a regimen that involves using only one expensive medication, that is in fact not often the case. It found that combination multi-drug cancer regimens are often far less expensive than traditional monotherapies. That novel, multi-drug combinations are often better financial values than traditional monotherapies, and that having treatment cost and value data at the point of care enables the prompt selection of more cost-effective medications and the avoidance of expensive low-value therapies that are clearly financially wasteful. While instructive, this is also lamentable, as it is likely that the mistaken presumption that combination therapies would be excessively expensive could have dissuaded some physicians and patients from pursuing a multi-drug option that may have been effective. According to Richard Nicholas, the study's author, few doctors are aware of the cost of the drugs they prescribe. Others have misconceptions about them. This often results in high-cost, low-value treatment. Our study shows why there is no reason to continue the practice of uninformed, high-stakes oncology prescribing, with the advent of healthcare AI, that can guide drug selection, to improve effectiveness, avoid waste, and drive value. This study's results have broad implications for the concept of medical necessity, and the practice of medical care management. CureMatch's clinical value is obvious. It helps oncologists become 350% more effective at identifying, evaluating, and selecting those drug treatment regimens that are likely to be most effective at defeating a tumor's known actionable mutations. It helps to drive better responses with matched treatments, selected sooner. At the same time, by enabling doctors to select the best financial value, it mitigates patient financial toxicity as well, by reducing their share of their treatment's cost. CureMatch also provides a physician, or medical care management nurse, the science with which to reverse past drug selection decisions that were based on sunk treatment costs, by identifying drugs that are simply not likely to be effective. Apart from its clinical value, CureMatch has a broad scope of utility for payers, plan sponsors, and their medical care management service providers. As evidenced by the case study examples, CureMatch can be deployed in a variety of payer settings, to eliminate wasteful spending on high-cost, low clinical value drugs, and to extend the value-based construct to include oncology specialty pharmacy, by facilitating physician use of multi-drug treatments that might otherwise not be considered. CureMatch can be used by prior authorization and utilization review nurses, to evaluate the medical efficacy and cost-effectiveness of various treatments, and to direct clinicians to therapies that are likely to be more effective and a financial value. It can be used to identify drugs that could create an adverse drug event, drugs that may be generally problematic, and the outlier providers who prescribe them. CureMatch reports can buttress coverage and prior authorization decisions, with science-based evidence. All of this helps to mitigate payer liability and risk. Lastly, CureMatch can provide support and guidance to oncologists who may want to try a novel multi-drug treatment, but don't want to deal with the tedious, time-consuming treatment approval process. CureMatch can be made a part of the prior authorization process and promote better provider relations, by simplifying treatment approval to be more consistent, science-based, and user-friendly.
There are a multitude of use cases for healthcare AI. As it relates to CureMatch alone, almost all of these healthcare stakeholders could deploy it, to improve health outcomes. reduce wasteful spending, extend value purchasing to oncology specialty pharmacy, and mitigate cancer risk and liability. As illustrated, CureMatch can be deployed by payers in a variety of ways, enabling them to be as passive or aggressive as they wish with respect to optimizing care, maintaining patient satisfaction, and driving value. As a practical matter, it is likely that payers and plan sponsors will first introduce CureMatch in an informational way, as an option, whereby the patient's provider is invited to have an NGS test and CureMatch analysis performed, at no expense to the patient, with the proviso that the treating physician may do with the results what they wish, with no penalty or added treatment or coverage restrictions. In this way, providers and patients can become familiar with how CureMatch works. Over time, if it is shown that CureMatch is effective at improving outcomes, mitigating wasteful drug purchasing, or driving value, it is likely that the program will morph into a process that becomes more of an evidence-based control measure. These scenarios are illustrated as implementation options B, C, and D, and characterized as judicious, aggressive, and intense. In these scenarios, NGS testing and a CureMatch analysis would first become incentivized, and then become more of a requirement. Likewise, incentives, penalties, and requirements may be built into the process to encourage, or even require, a physician to follow or be guided by CureMatch's recommendations, with the purpose being to not only improve outcomes, but to also mitigate waste and drive value. Drug selection options, in the form of incentives, limitations, and restrictions, may also be built into the process to ensure that plan assets are used prudently, fairly, and equitably, without waste. It has been suggested that, as an ERISA matter, a plan sponsor could be abandoning its fiduciary responsibility to the plan participant, by not covering the cost of NGS testing and a CureMatch analysis, knowing that it is effective at improving outcomes, and readily available. Some experts believe that a plan sponsor would be well within its rights, to reduce the universe of drugs to be covered for a certain patient as medically necessary, to only those that appear on a CureMatch report, the logic being that, to do otherwise, would be to waste plan assets on drugs that are not likely to be effective for that patient. Like other medical cost management and evidence-based utilization controls of the past, plan sponsors and payers will learn how to implement and utilize innovations like CureMatch, in ways that best meet their specific needs and desires. As promising as CureMatch is, the fact is that, on average, it takes a full 17 years for a new medical technology or healthcare innovation to get from the workbench into widespread clinical use. In large part, this is because the research required to prove medical efficacy and effectiveness differs from that which is required to be conducted to assess return on investment, cost effectiveness, and comparative value, measures that are of critical interest to payers. Translational research is undertaken with the intent of reducing the amount of time it takes to get a new medical technology, or healthcare innovation, adopted and diffused. Conducted in real-world settings, it provides payers with the financial information they need to justify adopting new technologies and innovations. As more and more translational research is conducted on healthcare AI-based innovations, like CureMatch, these solutions will become more widely adopted, faster.
These are the key takeaways of today's presentation. New, AI-supported precision medicine technology is proving to be a critical tool to improving health outcomes and patient satisfaction, while at the same time reducing waste and cost, and driving value. Coupled with tumor-specific molecular data, healthcare AI helps doctors choose the best course of treatment for cancer patients, and helps payers to extend value-based purchasing to cancer specialty pharmacy. Many AI-supported healthcare technologies represent high-value innovation opportunities for payers, not just based on their obvious clinical utility, but also based on their ability to help to reduce waste, contain cost, and drive value. Look for translational research and comparative effectiveness analysis, conducted in real-world settings, to help accelerate the adoption of emerging medical technologies and healthcare innovations, by all stakeholders.
Thanks for your attention. The purpose of this presentation was to help fuel your fire for professional knowledge, and to satisfy your career development needs with a cutting-edge learning experience that provides the latest need-to-know information. We hope it achieved its purpose.
Visit our website to download it in PowerPoint or PDF formats. If you have questions, or would like to express a comment, please contact us.